Molecular basis of life. Molecular basis of life is kind of misleading, right? Because, uh, as we talked about in the last couple of videos, couple of last lectures, we're talking about all sorts of molecules that make up our bodies. Our carbohydrates and nucleic acids and proteins and various kinds of lipids and other smaller molecules. So you would think that molecular biology would be concerned with all of those molecules. However, it's not. Molecular biology is primarily concerned with nucleic acids, DNA and RNA, and whatever DNA and RNA do. And then, tied to that is the genetics, how the DNA and RNA confer heredity, inheritance from one generation to another. So DNA is the focus of molecular biology. It is a long, double-stranded molecule as it resides inside of the nucleus of every cell. Remember, there's also, a little bit of DNA inside of mitochondria. It's a double helix that is 3D shape. It is usually tightly coiled, forming chromosomes, and histones and other proteins are protecting it from the outside. It's kind of packing those chromosomes so they cannot be uh, attacked by all sorts of other enzymes. And so the DNA inside of the protein envelope is kind of protected. Each of the two strands of the DNA molecule is a chain made of smaller molecule. Each link is in the chain is composed of a nucleotide. A nucleotide consists of one sugar molecule, which is deoxyribose in DNA or ribose in RNA, one phosphate molecule, and one nucleotide molecule that can be adenine, thymine, guanine, or cytosine. The two strands are uh, attached to each other in such a way that when you look at one strand, you know exactly how the other strand looks like because those four nucleotides are not promiscuous. Those four nucleotides can bind to each other only in certain ways. Adenine always binds with thymine. Guanine always binds with cytosine. You don't have any kind of cross-linking between those. So whenever you see an adenine on one chain, you know there's thymine on the other. Thymine on the first, adenine is going to be on the second. If there's guanine on one, cytosine has to be on the other. So you know what is going to be on the other chain. So those two chains are like mirror images or like a template or like if you have a stamp and what's is stamped on the paper. Uh, so that's actually very, very useful, right? Because in DNA replication, you can separate the two strands and make an exact copy of each one of them. Each one of them can be used as a template to build a second chain. And so you start with a double helix DNA, and you end up with two double helix DNAs that are identical to each other because each one of them has used one of the chains as a template to make the other one. Wherever one has an adenine, the other one is going to have a thymine. And so there are enzymes that can build all of that in the process called DNA replication, which happens every time a cell divides. The exact sequence of nucleotides on a strand is called the genetic sequence. When you add up all the genetic sequences of all the DNA on all the chromosomes, that's called the genome. Actually, technically, genome is a sequence of one set of each autosome plus X and Y chromosome. Each cell in the body has exactly the same chromosomes as it has all the same sequences that it has entirely exactly the same genome. Uh, there, yeah, that's fine when you're just born, but as we grow older, the years pass, there are going to be mutations as the cells in our body divide all the time. And so whenever there's a cell division and another one and another one, mutations can happen, which means that some cells are going to have slightly different sequences than the other cells. And at the end of life, pretty much every cell in your body has a tiny little bit different sequence than any other cell in the body. Most of those mutations mean nothing. They don't affect the function of the cell in any other way, but sometimes you might get a mutation that can lead to a tumor of some kind. So that's the genome. Now you are wondering, what is a gene? A gene is a small portion of the genome. It's a sequence of nucleotides, uh, from a couple dozen to a couple thousand nucleotides on part of the strand. And those nucleotides are expressed together. What does it mean expressed? They're copied into an RNA, uses a template to build an RNA molecule. So they're copied together as a strand. And then that RNA is going to have some function. There's several different functions of RNA, but the function that most biologists are most concerned about is the function of providing a template for building protein. Now, so cells use genes to synthesize proteins. This is a kind of a key role for DNA. If you look at the genome of like a human, a small proportion of it, very small proportion of it, actually are sequences or genes that code or are used as a template for RNA that is used as a template to produce a protein. Then there's an even smaller proportion of a genome that codes or is used as a template to build RNAs that have some other functions, from enzymatic uh, to transport RNAs to ribosomal RNAs. There are several different other types of RNAs, even smaller than the number of genes. The number of genes is only about 20,000. And about 90%, even more than 90% of our genome is what is called junk DNA. It has no function. It is a junkyard of old broken genes. It's a junkyard of viruses that some ancestor millions of years ago was infected with, and that sequence stayed there. There are repetitive sequences like a million TATATATATAs in a row. No function. There are scientists, biologists who are really sincerely trying to find a function for it, or just waving that there must be a function. But there's absolutely no reason that there is a function for all of that. It's quite correctly called junk DNA. We don't need any of that DNA. There are little bits of DNA that are functional without producing RNA, without being used as templates for RNA. Those are centromeres in the middle uh, of each chromosome that are tying two kind of branches to sides of the chromosome together, and there are telomeres at the very end of each chromosome, which are necessary for proper cell division. There are about 50 telomeres at the end of each chromosome, and each time there is a cell division. The very very end, the last telomere cannot be replicated, so the next division is going to have 49. The next division is going to, after the next division, you have 48, 47. After about 50 divisions, the DNA replication is going to encroach into the functional parts or the junk DNA parts of the chromosome. There are going to be errors, and that cell is going to die. So this is called a Hayflick number, because this guy Hayflick who came up with this, where the normal cell is going to divide only 50 times. It all gets reset when we make egg and sperm cells and fertilization. This also gets reset in cancer cells that can go on forever. You have enzymes that can replenish the deleted telomeres. Okay, so how does a cell use the DNA sequence to make proteins, which is like the most important function of the DNA? This is a two-step process. The first one is transcription, and the other one is translation. Transcription is when a sequence of a gene, DNA, is used as a template to make an equivalent RNA molecule. So remember, T and A, G and C always make always bind together. RNA is going to have uracil instead of thymine, very similar molecules. So A is always on the DNA is always going to result in U on the corresponding RNA molecule. C always makes a G, G always makes a C, and T always makes an A. So you build a molecule of RNA, which is called the messenger RNA. That messenger RNA is now template for building the protein. Now there's going to be some other modifications. There are introns, the little pieces of that sequence are actually cut out, and the rest of the sequence is glued together. So there's some modification of the RNA before it goes out to the nucleus and binds a ribosome somewhere embedded into the membranes of the rough endoplasmatic reticulum. There is that RNA is going to start winding around the groove in the middle of the ribosome, and it's going to serve as a template for building proteins. How? Each three nucleotides in a row are going to code for a particular amino acids. Remember that proteins are built of 20 essential amino acids. There are 20 different molecules that when you put them in a chain, you make a protein, a polypeptide or a protein. So RNA is going to code for like that. That's called genetic code. And you have a table in various books, and maybe even your book, I forgot, that gives you the exact sequences, the triplets, the three nucleotides in a row, which amino acid they code for. That is the genetic code. So some amino acids are used rarely, and they usually have only a single code, only a single triplet. Those that are used more often have three, four, six different combinations of nucleotides can code for that same amino acid, which allows for some mutations. You still get the same protein even if there's a mutation in DNA. So then there is the start codon, which is always methionine, and there's a triplet that codes. It's called a stop codon, which codes for nothing. There's no amino acid, so that's where the chain breaks off. So using transport RNAs that bring each one of those amino acids to the ribosomal RNAs that are part of the ribosome itself, and the mRNA that arrived from the nucleus, you have now set up the machinery to build a polypeptide. So that machinery reads the first three nucleotides. First is going to be methionine, and then the second triplet is going to code for a particular amino acid, and the next one, and next one, and next one. And so that chain of RNA is going to be used as a template for building a protein chain. The first process is called transcription because you're going from DNA to RNA, you're using the same language. Like, I'm talking and you're transcribing my speech is the same language. Translation is when you go from one language to another. Now you're going from a language of nucleic acids using the code, like a secret code, to build protein, which is a completely different language, the language of amino acids. So you end up making a polypeptide, which is a chain of amino acids. That's just the beginning of building a protein. The protein that you just got, the polypeptide that you just got, is a chain of amino acids, and that order of amino acids is important, but that's only what is called the primary uh, structure of the protein. That that polypeptide is built of various amino acids. Some amino acids are neutral, but many amino acids have a positive end and a negative end to it, which means they're polar. So positive and positive are going to repel each other, positive and negative are going to attract each other. And all of this is happening inside of solution, which means inside of a water. Water is a polar molecule. It has positive and negative ends. And so there's going to be a lot of this kind of electrochemical pushing and shoving around, which is going to change the shape of that protein in just according to the electrochemical properties of the amino acids in the water around it, which also changes in different pH. What you're going to get is what is called a secondary structure. So, and kind of just physical forces, electrochemical forces modify the shape of the protein that is the secondary structure. That structure, the protein still doesn't make the protein functional. That structure is not enough for the protein to be able to do its job. So here come other proteins, those are enzymes called chaperonins, like a chaperone at a dance, right? So they come in and they modify the 3D structure of the protein. So they fold it in particular ways. They make spirals and sheets and various parts of it in a particular shape. After all, it's the 3D shape of a protein that gives it its function. Because often it has to, lock onto, like, key a lock onto some other molecule in order to perform its function. So the tertiary structure, the one that is built by chaperonins, is what make a protein into a functional protein. So all that folding is necessary for the protein to have its function. You may have heard of molecules called prions. They cause diseases like mad cow disease and scabies and Creutzfeldt-Jakob disease. Those are proteins. And those proteins have the primary structure, and because of that, the secondary structure, the same as healthy proteins in our bodies, mostly like in our neurons, in our brain cells. So if by some, big grave mistake of your life, you have eaten nervous tissue of a cow with mad cow disease or a sheep with scabies or something like that. Those are proteins that have the primary and secondary structure exactly the same as your healthy proteins. So your antibodies and all that stuff, they're not going to recognize it. It goes straight into your brain, and the healthy version of that protein and the prion version of that protein meet each other. What's the difference between them? They have the same, pretty much the same order of amino acids, so they have the same primary structure. They're inside of the same kind of liquid water, and they have the same order of positive and negative and neutral amino acids, so they have roughly the same secondary shape, but they have very different tertiary shape. A healthy protein has a tertiary shape that was modified by chaperonins. The prion has a very different shape. What's the problem with the prion is that it can act as a chaperonin itself. What is it going to do? It's going to find its equivalent protein. It's going to reshape it from a healthy version to its own version, to the sick version. And so you started with a healthy protein and its equivalent prion, and then the prion reshapes this one in seconds or minutes or hours. It can be a long process, and you end up with two prions. And then you end up with four prions, and eight, and 16. It might take years, but in the end of that, your brain is gone. Your brain cells are dead. So tertiary structure is very important for the proper function of the body. If your 3D structure of the proteins get misshapen, then they're just sitting there glutting in the cells, taking all the energy and material and space in the cells until the cell dies. We're not going to stop at the tertiary structure. There's more things that happen to proteins after the chaperones are done with them. And that is adding various things to them, and often adding multiple proteins together, binding them together so they make a larger structure. For instance, hemoglobin is made of four subunits. There's, gene 1, gene 2, each one of them makes RNAs, each one of them makes a protein. So you have subunit A, subunit B, you get two copies of subunit A, two copies of subunit B. You put them together, you have hemoglobin, which has four subunits, two A's and two B's. Each one of those subunits also gets another molecule added to it called heme. And then each heme gets added an iron ion to it. So all of that together, so binding together different subunits, adding other small molecules to it, maybe ions to it. That is called quaternary structure of the protein. Pretty much every protein is also going to have a small chain of carbohydrates added at one end. That chain of carbohydrate is called a zip code. It tells the cell exactly where to transport, using cytoskeleton, where to transport that protein, where to store it inside of some vesicle or Golgi apparatus or wherever, or to take it out of the cell or embed it into the membrane, exactly where that protein is supposed to go once it's synthesized and modified from primary to secondary to tertiary to quaternary structure. Okay. So this is kind of the basis of molecular biology. We went briefly through DNA replication, DNA transcription, DNA translation, see what else the book says about it. Uh, then the next thing is uh, the cell division. So the only way to build a cell is by dividing an existing cell into two. Yeah, they have made artificial cells in the lab recently. That's us. But in general, in nature, the only way to have a cell is if a cell divides into two. That also requires the genome, the complete sequence of the DNA, which is an essential part of the cell. It needs to be duplicated. So you have one in the one daughter cell, one in the other daughter cell, and that is DNA replication. So I'm not going to ask details about DNA replication. This is not a molecular biology or genetics class. We have 23 pairs of chromosomes. Each chromosome gets duplicated. All of the sequence gets duplicated. Each daughter cell gets one copy. So once the DNA is all replicated, all the chromosomes are replicated. One set goes one side, one goes to the other side. A membrane gets built in between, and then along that membrane, the two cells can separate from each other. And that's how it's split. So what I just described is the simplest cell division, which is called mitosis. Mitosis is how all the cells are duplicating itself. I mean, that's the essence of growth. That's the essence of repairing dead cells and replacing dead cells. So, what you get is you start with a diploid genome, duplicated, and each one of the cells gets a diploid genome. The big exception are the gametes. The gametes are egg cells and sperm cells. What would happen if they divided by mitosis? You have a diploid egg cell with a full complement of all the chromosomes, and you have a diploid sperm cell with a full set of chromosomes, the entire genome. And then they fertilize, they fuse together. What do you have? Four, not two. Next generation, eight. Next generation, 16 genomes, copies of the genome, right? After a while, you have an enormous cell packed with DNA, taking all the energy to take care of that DNA and then to replicate it and to build another one. Oh my. Now, we've used some of that. That's called polyploidy. A lot of fruits and vegetables are polyploid because having multiple genomes enlarges the cell, which means there's more water in the cell. Because the cell is bigger, there's more sugar. There's less membrane, right? So you have this juicy watermelon instead of a horrible little watermelon of the past. So it is necessary to divide gametes in a different way. And that different way is called meiosis. Meiosis, or the reduction division. It reduces the genome, right? So you start with a diploid cell and you end up with a haploid cell, which has only one copy of the genome, not two. So meiosis has a couple of different stages: meiosis one, meiosis two. We're not going to go into details of that. All these anaphases and prophases. This is part of general biology and genetics courses. Remind yourself of that if you forgot, but I'm not going to ask that. What I'm interested in is this logic of those whole things. You need to have haploid gametes because once they fuse during fertilization, you end up with a diploid cell. The diploid cell can then divide into more and more cells, producing an embryo. So meiosis is essential for reproduction because it produces haploid gametes, haploid eggs, and haploid sperm cells, which are going to produce a diploid zygote. If that didn't happen, all the cells would be polyploid, and after several generations, this line would end. That organism would have so many mistakes, and few very enormous cells that are using all of that time and energy trying to maintain enormous quantities of DNA, and everything else would grind to a stop, and that organism would die. Probably embryonic development would not work well because there's just too much DNA, which means too many sources of error, and all sorts of things would go wrong. Okay, uh, so once you have that cell uh, formed, the zygote formed from a haploid sperm cell fertilizing a haploid egg cell, you get a diploid cell, which is a zygote. It is a totipotent cell. It's a totipotent cell, which means that it can become any cell type in the body. There are more than 200 types of human cells: nerve cells, muscle cells, skin cells, all sorts of different kinds of cells, pancreatic cells, A, B, and alpha, beta, gamma. There are all sorts of, there's 210, 220 different types of cells in human bodies. And the zygote can become anything. It's a totipotent cell. So the zygote starts dividing into 2, 4, 8, 16. And as it divides, it's going to uh, need to make different cells different. So you have a zygote, which is not a nerve cell, it's not a muscle cell, it's all of them all at the same time. It's just a sphere. It doesn't have function of any one of those cells, and yet has a potential for all of those functions because it has the entire genome. So as it divides, those cells need to differentiate from each other. Cell differentiation. What is cell differentiation? It's the change of which genes are turned on and which genes are turned off. What does it mean turned on? DNA is twisted, covered with proteins, nothing happens there. Turned on, that's turned off. Turned on, untwisted, uncovered, the transcription molecules can start the transcription of that piece of the DNA, which means that gene can be transcribed and translated. Uh, and different cell types differ from each other in which genes are turned on and which genes are turned off, which are expressed, which are not. Uh, a muscle cell needs genes that are essential for muscle function. A nerve cell needs to turn on genes that are necessary for making neurotransmitters and neurotransmitter receptors and those kinds of things. So different cells are going to turn on and turn off different genes. This is all happening during the embryonic development, where you can have a gradient of various molecules in the cell. The fertilized egg cell has polarity, has a north and south pole. And in different organisms, it's different. Sometimes it's really what's up and what's down, gravity, like in plants. It can be exact spot where the sperm enters the cell. But in most, like mammals, like us, what is determined by polarity is where, at the end of meiosis, where does the egg cell, that uh, haploid cell, where does it store a whole bunch of already transcribed RNAs? So a bunch of already transcribed RNAs are sitting in one corner of the cell, and that's the North Pole. As that zygote starts dividing, some of the cells are going to contain those RNAs and others are not. As those cells start translating those RNAs into proteins, those proteins are going to act on DNA. They're going to act on DNA, turning on or turning off various genes. So the cell that has some of those RNAs is going to start producing some molecules, some proteins, and not some others. Some of those proteins in these early days of the embryo can, easily pass from one cell to another, to another, to another. And so you have a concentration gradient, the greatest at the North Pole and almost nothing at the South Pole. And that level, the concentration of within that gradient, the concentration of that molecule is going to have different effects on different cells. Depends on how far away it is from the North Pole, turning on or turning off different genes. So you end up having a whole bunch of different cell types right there from the beginning that are differing from each other in their patterns of gene expression. Which genes are turned on, which genes are turned off. Once you get to that point, when you take those cells out, for instance, and see what happens, those cells are now not totipotent. They have modified their gene expression patterns in such a way that they are uh, multipotent or pluripotent. They can become a lot of different cell types, but not all of them. As the zygote keeps dividing, cells keep dividing, it starts moving around, making a hollow tube. That hollow tube is going to have three layers: the endoderm on the inside, the mesoderm in between, and the ectoderm on the outside. Each one of those layers is pluripotent. It can become a particular types of cells and not others. The ectoderm is going to produce nervous system, skin, those kinds of things. Endoderm is going to produce bones, muscles, um, not muscles, bones, GI tract, respiratory tract. And mesoderm is going to be cardiovascular, muscular system. There is, of course, some exceptions. Some cells go back and forth. So you're going to have three layers, and each one of them is going to be pluripotent in a different way, in a way that allows them to become a large number of different cell types, but not all 210. So there's already a reduction in how many cells a cell can turn into. Um, so the pluripotent cells of the endoderm, ectoderm, and mesoderm, they keep growing and dividing and differentiating into different cell types. Those are pluripotent. As they differentiate into more and more cell types, those cell types are now multipotent. They're not pluripotent anymore. They're multipotent. They can become only a very limited number of cells. So, for instance, you're going to have a stem cell that can become 8 or 9 different types of blood cells. Can't become anything else. Another stem cell can only become, several different types of nerve cells, nothing else. So that's now not a pluripotent stem cell. This is now a multipotent stem cell. And once that divides, it makes a final version that is unipotent. It can be, it's itself. When it decides, once it divides, it can only divide into a copy of itself. So these diffusible chemicals that go from one cell to another and induce you to change the expression of genes, some turning off, some turning on, and turning them thus into a different cell type, that process is called induction. So the best example of induction is how the eye develops. So retina of the eye is an outgrowth of the brain. So you have an embryonic kind of head, which has an embryonic skin around it. And inside of that head, there's an embryonic brain developing. And out of that brain, kind of go out eye stalks and, nerves, which is going to be an optic nerve and a retina. And that retina then goes on the inside of that skin, developing skin of the embryo, and that developing retina is going to secrete chemicals that are going to affect those skin cells right on top of it. You're going to transform those skin cells into things like cornea and ciliary body and iris and uh, pupil and all the other parts of the eye. So that is called induction, when molecules secreted by one cell or layer of cells or a bunch of cells are changing the gene expression in neighboring cells, turning them into a different cell type. Some cells migrate during the embryonic development. For instance, the best example are the cells of the neural crest. The neural crest develops on the kind of, this is the embryo, you have a neural crest on top of this. A lot of things develop there, that's the beginning of the nervous system, the spinal cord. But neural crest cells are kind of the crest, are the cells kind of on the sides of that ridge. That's going to become spinal cord. And those cells migrate all over the developing embryo. And what they're going to become is dependent on the neighborhood where they end up, because there's going to be induction there, right? The neighboring cells are going to induce them to become different things. So if they go to the skin, they're going to become melanocytes, carry the color, right? They carry the pigments that give the skin its color. If they go, for instance, to end up in the like adrenal medulla, to the adrenal area, they're going to become the cells of adrenal medulla. There's going to be adrenal cortex around them. So wherever they go, they're going to become something different. And that is very, very interesting, because the rate, the speed at which they migrate is going to have an effect. And this is what's happening with domesticated foxes. I don't know if you heard of that experiment. It started in 1960s in Siberia, uh, where they started breeding foxes for behavior, only behavior. They were breeding foxes that are specifically friendly to humans, and they're breeding foxes that were very, very aggressive and unfriendly toward humans. So two different strains, and those became very, very different from each other very, very fast, or very few generations. It's been going on since the 60s. There's been 60, 70 years of it now. Um, so 60 years definitely of the experiment. And so what's happening is that friendly uh, those friendly foxes are changing all sorts of other things. Suddenly they have floppy ears, curly tails, white paws, white noses. Uh, their snouts get a little shorter. Uh, they start breeding twice a year instead of just in spring. They start barking. There are other behaviors, more and more like dogs. And this is called, kind of a domestication syndrome. If you think about, cows and pigs and horses and bunny rabbits and all sorts of things that have been domesticated, a lot of them have funny colors and, white legs and white tips of tails and floppy ears and a star on the forehead or something like that, or piebald kind of painted coloration. And where does it come from? So it comes from selecting for animals that are less stressed by the presence of humans. Remember, that was selected only for their behavior towards humans and nothing else. So that led to the selection for the animals that produce less stress hormones. Where do the stress hormones come from? From the adrenal medulla. Does adrenal medulla come from? From the neural crest cells in the early embryo. So what all of that selection has actually done is selected for the delay of the onset of migration of these cells. So the neural crest cells start migrating later. So it's acting on gene that times the start of that migration. And so those neural crest cells don't get to the area of the adrenal soon enough, and so there's only a few of them, and the medulla is not as big and doesn't produce as much adrenaline. It's not as much stressed. It doesn't get to the tip of the tail or to the paws in order to produce melanocytes, so those parts remain white. It doesn't get to the tips of the ears where it will become cartilage, so floppy ears. So all sorts of things. Wherever neurocrest cells go, they are going to delay and they're not going to be able to do their job at all or not as much. And that explains the entire change in domestication of foxes. So migration is very important. Exactly which cells migrate, where do they go, and when do they go there? Um, there is another aspect of the embryonic development, and that is the programmed cell death. It's called apoptosis. So during development, often the embryo produces too many of a particular type of cell and then induces them to kill themselves if they're extras. That often happens by induction. This happens by apoptosis. The cells of the developing fingers are inducing the neighboring cells in between them to kill themselves. So we start with a pedal and end up with fingers and toes. You disrupt that process, you end up with flippers in dolphins and whales. You end up with wings in bats. They all started with five fingers, but that apoptosis of the stuff between the fingers, if it stops, you can evolve something like a fin or a wing, which sometimes, occasionally, by mistake, happens in a human embryo. It's born with flippers. So apoptosis is an important process. It happens in the brain. The brain, or developing brain, makes way too many nerve cells, and they follow various gradients of various growth factors, and spread around, and they start making connections with each other, the synapses with each other. If the synapse is used, which means if that cell tells this cell what to do and it's useful, right? Because you're sensing something in the environment or you're moving a muscle or something like that, then that synapse strengthens. If that synapse is not used, there's no need for it. No sensory or motor activities happening through that connection. That synapse is going to break off. If that cell now has nothing else to do, it kills itself. It's apoptosis. The target cell now that has nothing else to do is never used for anything. It kills itself. So there's pruning of all the brain cells during the development through the process called apoptosis, a programmed cell death, a series of cascade of kind of genes turning on genes, turning on genes that are resulting in the cell destroying itself. Okay, so that embryo, when it grows up to be a fully grown organism, is going to resemble its parents more than it resembles other individuals in the same population. This subdiscipline of biology that studies inheritance is called genetics. and is interested in how various traits are inherited from one generation to another. For them, it's also important to understand what a gene is. The way we described the gene a few minutes ago was that it's a sequence of DNA, so a bunch of nucleotides, a couple dozen, couple thousand, a couple hundred, doesn't matter, that are transcribed as unit to make an RNA that is going to have a function of some kind, either making a protein or doing some other function in the body. But for studying inheritance, you don't need to know what is the chemical basis of a gene. That is what is called a conceptual gene. A conceptual gene is a unit of inheritance. Gregor Mendel in 19th century, when he discovered the basic principles of genetics, had no idea what was the chemistry underneath. But he could develop the basic rules of inheritance, things like dominance and codominance and recessiveness of a trait by breeding peas and figuring out how the traits are inherited. So the conceptual gene is still useful. Population genetics and quantitative genetics are mathematical sciences that are studying inheritance of traits, and it doesn't matter if those traits are coded by genes made of DNA or made of whatever. And on some other planet, it might be something else. And yet, the rules of population and quantitative genetics would stay the same for an alien life form that doesn't use carbon, doesn't use DNA. If there's inheritance, it obeys the laws, and in which case we are talking about conceptual genes and not the material genes that are made of DNA on this planet. Okay. So when you're talking about the genes, remember that we have a genome that's diploid, so we have two copies of every chromosome, which means we have two copies of each gene. Sometimes those two copies are identical, but usually there's some difference between them, so we have two different alleles of that gene. Sometimes that difference between alleles matters. For instance, one of those alleles, if you have one of those alleles, let's say you have two different alleles, one of them might be resulting in a trait while the other one is silent and not doing its job, which means this one is dominant and this one is recessive. And your kids are going to have, some of them are going to have one, the other one are going to have the other, and depending on what kind of genetic environment those find themselves, the kids are going to exhibit the dominant trait or the recessive trait. Sometimes they're equal partners, and you get something in the middle. You get codominance, where the two genes are each contribute to something to the trait that either one of them alone could not do. If you have same alleles for the gene, you're homozygous for that gene. If you have two different alleles, it is called heterozygous for that gene. Some genes are not so independently moving from one generation to another because they may be linked. Usually, the link is physical. The genes are right next to each other on the chromosome. So, even after crossover, they're less likely to be separated from each other. They're most likely going to go together to the next generation. So, sometimes you have traits that are going together with each other from one generation to another, and very rarely get broken apart. Epistasis means that an allele produces different traits depending on what kind of genetic environment it finds it. What are the other alleles there? So there are alleles A, B, and C there. The gene X is going to produce a particular type of trait. If it's F, E, and J, then it's going to produce a different type of trait. It's the same gene. Pleiotropy means that a single gene affects multiple traits. A lot of developmental genes, a lot of transcription factors are going to have many, many functions during the development of the embryo, and thus they're going to be involved in the building of many, many traits. Uh, Epigenetics is a process of inheritance that does not involve changes in the sequence of DNA. Everything from birdsong, which is learned from what generation to another. Two, let's say methylation DNA, thus affecting the expression of genes from one generation or another. Usually lasts just a few generations because the sequence of DNA is essential for long-term multi-generational inheritance. Okay, and I'm